Hi, welcome to my presentation on Chapter 6, Lipids and Membranes. So in this chapter, we're going to go over the fourth type of macromolecule that's found in organisms, which is the lipids. The main structure that lipids form in the cell is the cell membrane. So we'll also talk about the cell membrane at the same time, kind of, as we talk about the lipids. So another name for the cell membrane is the plasma membrane. The membrane is two layers of lipids that basically form a ball around the cell to separate that cell from its environment, or to separate the contents of the cell from its environment. By having a membrane, the cell gains a lot of advantages. For one thing, it's able to kind of keep a lot of toxins out of the cell and a lot of nutrients in, and just basically reactants for all the different chemical reactions that need to occur in the cell. One way to make a reaction go faster, as we learned in the chapter on enzymes, is just to increase the amount of substrate available, and the substrate would be the reactant for a reaction that an enzyme will catalyze. So if you're able to kind of concentrate those reactants in a small area, then you're going to raise their concentration and increase the rate of reactions that use them. So the cell membrane is able to kind of do that by making sure that reactants stay inside the membrane and can't get out of it. The diagram here is kind of a picture of a cell membrane. Well, this here would be a picture of a cell membrane. So you can see that it's a ball made of two layers of lipids. And if you zoom in on the membrane, you'll see those two layers. So you have the top one and the bottom one. These lipids all have kind of little balls at the top of them and then tails coming off. And if you zoom in even further on one of those lipids, you would see it looks kind of like this. And we're going to go into the structure of those lipids in a bit more detail later in this chapter. So a lipid is kind of loosely defined. It's just any organic molecule that is hydrophobic. So any molecule that is based on carbon and does not dissolve in water is going to count as a lipid. So as you might figure, there's a lot of different types of lipids around, and they're not necessarily all that closely related to each other in terms of their structure, beyond, of course, all being based on carbon and not being hydrophilic. In order to be hydrophobic, a molecule has to have a lot of nonpolar bonds in it. For organic molecules, that generally means you have a lot of carbon and a lot of hydrogen. Since those types of atoms have about the same electronegativity, when they bond to each other, they always make a nonpolar bond. So anytime you have a carbon bonded to a carbon, that's a nonpolar bond. Anytime you have a carbon bonded to a hydrogen, that's also a nonpolar bond. There's several major types of lipids that are kind of commonly encountered that we're going to go over. It's not an exhaustive list of the types of lipids. There's a lot of different types. There's just some of the kind of more common or more important ones. So the first one we'll go over is the hydrocarbon. A hydrocarbon is a really, really simple type of lipid. It's just nothing but carbons and hydrogens. So any molecule that contains only carbon and hydrogen can count as a hydrocarbon for the most part. A hydrocarbon, I guess the place you would most commonly encounter them is actually in like gasoline. And up here you have a diagram of a hydrocarbon. So it's just a chain of carbons, and they have hydrogens attached to them, and then sometimes there's other carbons attached as well, kind of coming off like a branch, but basically just a chain of carbons with hydrogen stuck onto it is a hydrocarbon. A fatty acid is slightly more complicated. It's a hydrocarbon that also has a carboxyl group. And the carboxyl group involves oxygen atoms, so it's not just carbon and hydrogen in the fatty acid. Over here we have examples of fatty acids. So you can see the hydrocarbon part of it, just a chain, just a chain of carbons with hydrogen stuck on. Over here it's the same thing, chain of carbons with hydrogen stuck onto it. And then at one part of the molecule, or at one end of the molecule, you have the carboxyl group, which makes it the fatty acid, because that carboxyl group is slightly acidic. There's two basic types of there's two basic types of fatty acids. One is saturated and the other is unsaturated. The straight molecule here is a saturated fatty acid, We say it's saturated because every carbon is bonded to the maximum amount of hydrogens that it could possibly be bonded to, given that it's participating in this chain. So it's kind of saturated with hydrogens. In order for the molecule to be like that, all of these bonds between the carbons have to be single bonds. And when you have the molecule built out of just single bonds like that between the carbons, that gives you a straight molecule. Over here you have an unsaturated fatty acid, which has mostly single bonds between the carbons, but these two carbons here are attached by a double bond. Every time you have a double bond in the molecule, it introduces a kink. So that's why the molecule bends at that location. It's forced to bend by this double bond. So an unsaturated fatty acid is always going to have some bends in it like that. How many bends it has is going to depend on how many double bonds it has. It could have just one, or it could have multiple. And when you have these carbons double bonded to each other, you notice that they are now attached to fewer hydrogens than the average carbon in this molecule. So for these double bonded carbons, they're only bonded to one hydrogen atom. The other carbons that are attached by single bonds are each attached to two hydrogen atoms. So that's why we call it unsaturated, because it doesn't have the maximum amount of hydrogens that the carbons could be attached to. So a little bit more about the saturated versus the unsaturated fatty acids. The fact that saturated fatty acids are linear ends up being kind of important for their physical properties. Because they're just a line, you can actually line them up together and pack them in together really tightly. And when they're all tightly packed together like that, then it's kind of more difficult to melt them and turn them into a liquid. So saturated fatty acids are more likely to have a high melting point or a higher melting point than an unsaturated fatty acid would have, which for practical terms means that when we have that type of fatty acid at room temperature, if it's saturated, it's more likely to be a solid at room temperature. If it's an unsaturated fatty acid, then it has those bends in it, at least one bend, and those bends are going to prevent the acids from packing together too tightly. So there's going to have to be some spaces in between them when they try to pack together. And those spaces mean that they end up having a lower melting point than the saturated fatty acid would have, which practically means that when you have that type of unsaturated fatty acid at room temperature, it means it's more likely to be a liquid. Then we have a couple special types of saturated and unsaturated fatty acids. For the saturated fatty acids, we have waxes. A wax is a type of saturated fatty acid where it has a really, really long chain. And the fact that the chain is so long and that it's straight so you can get them all packed in real tightly together, that makes it really, really hard to melt the waxes. So they have really high melting points, and when you have them at room temperature, they're not just solid, but they're very kind of stiff and hard to manipulate. Then for unsaturated fatty acids, the kind of special type is an oil, which is just any unsaturated fatty acid that is a liquid at room temperature. So there are some unsaturated fatty acids that would be solid at room temperature. It's just kind of less likely for them compared to the saturated fatty acids. And any one that would be liquid would be an oil. So you just have some examples here. On the top we have a substance, butter, which is composed mostly of saturated fatty acids. Joined together to form saturated fats. So they all have this straight chain, they're all able to pack in together really tightly, and that's the reason why butter is a solid at room temperature. Over here, on the other hand, you have an oil, and you can see that there's a couple of double bonds in the unsaturated fatty acid of that oil, which means that when you try to pack these molecules in together, they can't get that close to each other because they're held apart by these bends that they have. Which means that they end up being a liquid at room temperature and therefore counting as an oil. And on the bottom here you have an example of a wax. So you can see that the chain is straight, similar to butter, but it's just way, way longer than the butter chain. It's like 2 or 3 times, well, over 2 times, but maybe even 3 times as long as the chain of the saturated fatty acids in butter. Which means that when you have this type of saturated fatty acid at room temperature, it's going to be really stiff. And it might form a structure kind of like beeswax, which is quite solid at room temperature and hard to manipulate at room temperature. Some other types of lipids are steroids, or another type of lipid is a steroid. A steroid, there's lots of different ones actually, but all of them, what they'll have in common is that they have 4 rings that are made out of carbons, and then they can have different stuff kind of attached to those rings, like different chemical groups attached to the rings. Over here you have an example of a steroid. So you can see the 4 rings here highlighted in orange. These are made out of carbon atoms, and then they have different stuff attached to attached at different places on those rings. So depending on which steroid it is, it will have different stuff attached in different places, but it's always going to have those 4 rings at the center of it. There's actually a lot of steroids in your body that end up being pretty important for you. First you have cholesterol, which is a component of your cell membranes actually, and we'll talk in a bit when we go into more detail on the membrane. We'll talk about what cholesterol is doing in your cell membranes, but also a lot of hormones in your body are steroids. So for instance, estrogen and testosterone, which are the sex hormones in women and men respectively, those are both steroid hormones. They're actually derived from cholesterol. You also have cortisol, which is a stress hormone that's also derived from cholesterol. Yeah, so you have a number of hormones in your body that are actually steroids in terms of their structure. Another type of lipid, the type that you're probably most familiar with, is the fat. A fat is actually 3 fatty acids that are all attached together by a glycerol molecule. And we'll look at the structure of that in a moment. Another word for a fat is a triglyceride. The glyceride part because it has a glycerol, and the tri part because it has 3 fatty acids attached to that glycerol. So if you ever see the word triglycerides, that's just a fancy word for a fat. A fat could either be a saturated fat or an unsaturated fat. It depends on which fatty acids are kind of composing it. So if it's got all saturated fatty acids in it, then it's a saturated fat. If it has some unsaturated fatty acids in it, then it's an unsaturated fat. The main role of the fat in cells is just to store energy in the long term. And then the last major type of lipid that we'll talk about is the phospholipid, which is very similar to a fat, but a little bit different. So it's also got fatty acids attached to a glycerol molecule, but instead of 3, it only has 2. And actually, they technically could be hydrocarbons as well. So if the kind of chains that are attached to the phospholipid have a carboxyl group, then they are fatty acids. If they don't have the carboxyl group, they're just hydrocarbons. But you're going to have 2 of them attached to the glycerol molecule, and then you also have another thing attached to the glycerol molecule, which is going to be a phosphate group, and then attached to that phosphate group you have something else, some other organic molecule. There's a lot of different types of phospholipids that would have different types of organic molecules attached to their phosphate group. Phospholipids are really important for organisms because they're the primary component of the cell membrane for most organisms. Not for all actually, but for most of them. In eukaryotes and bacteria, the fatty acids that we have in our cell membranes are all in eukaryotes and bacteria, the phospholipids that we have in our cell membranes are all containing fatty acids and not hydrocarbons. But there are some organisms that have hydrocarbons in their membrane phospholipids. So here you have just a comparison of the structure of a fat versus a phospholipid. This is a fat. So you have 3 fatty acids here. You can tell they're fatty acids because they have the carboxyl group, and then it's a chain of carbons. Anytime you see something drawn like this in chemical structures, that means that it's made out of carbons and there's hydrogens attached. So just at each bend, that's a carbon atom, and then not shown would be hydrogen atoms, but each carbon would be attached to the maximum amount of hydrogen atoms that it's possible for it to be attached to. Then you have the carboxyl group at the top, and then at the very top of this molecule you have the glycerol up here. So the glycerol has 3 carbons with hydrogens attached to them, and also on the other side each carbon has 1 oxygen attached, which is where the fatty acid is going to end up binding to the glycerol. Actually, when you just have a free glycerol, it has hydroxyls, not oxygens. But you don't really need to know the structure of glycerol, just that it is there in the fat and that the 3 fatty acids are attached to it. Then over here you have a phospholipid. So here you have your glycerol, and you have 2 fatty acids coming off of it, not 3. The last carbon in the glycerol in a fat would have another fatty acid attached to it, but in a phospholipid it has the phosphate group attached to it instead. Then on the other side of the phosphate group you have something else, some type of organic molecule. In this case it's a choline molecule, but it could be different things depending on what type of phospholipid we're talking about. The phospholipid ends up being really useful for making membranes out of because it has the tail of it here, which is hydrophobic, and then the head over here is actually hydrophilic. You can see that the nitrogen in the choline has a charge on it. Also, the oxygen in the phosphate group has another charge. And you also just have a bunch of polar bonds in here with like oxygens attached to carbons, oxygen attached to carbon, or nitrogen attached to carbons. Those are all going to be polar bonds. So those are all places for water to interact with this part of the molecule. And in a moment we'll talk about why that helps the phospholipid form a membrane. So there's a lot of different functions that lipids serve in this cell. One of the primary ones, or I guess I should say the primary one, is just to make up the plasma membrane itself, which kind of, separates the cell from its environment and defines the cell in a way, kind of gives it, its identity as a discrete unit. The types of lipids that you find making a plasma membrane would be the phospholipids and also cholesterol. Actually, some organisms don't have cholesterol in their membrane, but they would have a different type of steroid. So they might have something else, not cholesterol, but something to serve that role. Something that's similar to serve that same role. The other, or I guess I should say the second most common function of the lipid is just to store energy. Lipids are really good at storing energy because almost all of the bonds that they contain are nonpolar. So in the previous chapter we had mentioned that carbohydrates are good at storing energy because they have a lot of nonpolar bonds, a lot of places where carbon is attached to carbon or carbon is attached to a hydrogen. Those bonds are longer, which means that they have more energy in them. But in a lipid, it's not just a lot of the bonds that are nonpolar, it's actually most of them, even sometimes all of them. In a hydrocarbon, all of the bonds are nonpolar, which means that all or most of the bonds are quite long and therefore store a good amount of energy in them. Then you have some other functions of lipids in the cell. There's some lipids that are able to kind of absorb photons. And we would call those pigments because they're generally going to have a color to them of some kind. There's a couple of major examples from the human body, which are retinol and melanin. Retinol is actually found in your eyes, and you need it in order to see. And then melanin is in your skin. It gives you your skin color, and its function would be to absorb UV light and protect your cells from that UV light. Lipids can also function as kind of messenger molecules or as hormones, so that would be actually the steroid hormones that we kind of mentioned, the estrogen, testosterone, and cortisol. So those are molecules that are released into the blood and will travel across the whole body and carry some type of message to a bunch of different cells in the body, in some cases all of the cells in the body. Another function of lipids is to keep water inside an organism. So your skin is full of lipids. And when you put lotion on, you're actually kind of replenishing the lipids in your skin. The lipids there are really important because they make sure that your water stays on the inside and it doesn't just evaporate to the outside. Right, your body is mostly water by volume, so you have a lot of water to lose, a lot more than is in the environment, which is generally someplace dry, unless you're actually living in the water, which as a person you are not. So it's really important to have that barrier in your skin that will not allow water to cross it. And of course, water can't cross the lipid barrier in the skin because those lipids are hydrophobic and water cannot interact with them. So it's just going to stay away from them. So you have that in, in humans, your skin is full of lipids, but also in plants, they will generally have a coating of lipids on their leaves. For some plants, it's kind of a really thin coat, so you don't really notice it, but for other plants that have a more waxy looking leaf, it's a very thick coat made out of actual waxes. And so it allows the plant to kind of pretty aggressively keep the water inside of its leaves and not just lose it throughout the day. And finally, another function of lipids would be to serve as vitamins. So certain vitamins are actually just lipids. Vitamin A is just retinol, actually. So that's of course important for your vision because it allows you to absorb light in your eyes. But it's also actually important in your kind of early childhood for your growth and development. You also have vitamin D, which is necessary in order to absorb calcium from the gut. Vitamin D is actually synthesized in the skin. It is a lipid, and if you don't have it, then you're actually not able to absorb calcium from your gut. So even if you're like drinking a lot of milk or something that has a lot of calcium in it, it would just be kind of running straight through you if you don't have vitamin D. Then you have vitamin E, which is an antioxidant. So kind of later in the semester, we'll go over cell metabolism and how it is that cells are capturing energy from glucose and storing it as ATP. As a part of that process, you're always going to generate certain molecules that we call reactive oxygen species, and they're just, they're very reactive. They have an oxygen atom in them, and they basically will react with almost anything that they come across and damage that thing. So whether it's enzymes or DNA, it's going to react with it and damage it. So an antioxidant is something that's designed to protect you from those reactive oxygen species. Basically, the idea is that the reactive oxygen species would react with the antioxidant instead of with something else, therefore not damaging the other parts of your cells. So you do generate those types of molecules through your normal metabolism, but of course you can also be exposed to them in other ways. If you're exposed to radiation, for instance, or certain compounds that you might ingest could be oxidants that would be just very reactive and have a tendency to react with enzymes or DNA and damage those things. And the antioxidants are protecting you from that. So that's what vitamin E is doing for you. Then finally, you have vitamin K, which is necessary in order for you to have proper blood clotting. So if you don't have vitamin K, then you will have a clotting deficiency, and if you get cut, you're going to bleed for a long time. So now we've gone over some basics about lipids, we'll turn our attention back to the plasma membrane, or the cell membrane. And as I had mentioned, the cell membrane, or the plasma membrane, is based on phospholipids. That's the main type of molecule that you find in a cell membrane. So in a phospholipid, you have kind of the two ends of the molecule. You have the head of it, and then you have the tail. The head includes the glycerol and the phosphate and the other organic molecule, maybe a choline. The tail is your fatty acids or your hydrocarbons. So that means that the tail is always going to be hydrophobic because it has almost exclusively nonpolar bonds. And then the head is actually going to be hydrophilic because it has some amount of polar bonds in it, and it also will have some charges. The phosphate is always going to have a negative charge on it, and then the organic molecule is usually going to have a charge as well. So in the diagram here, you can kind of see that the tail region is just the fatty acids. In this case, they're shown as fatty acids because they have the carboxyl group. If you didn't have this carboxyl group there, then they would just be hydrocarbons. So these are all nonpolar bonds, with the exception of the bond between oxygen and carbon up here in the carboxyl. The rest of these bonds are all nonpolar. So this is kind of the hydrophobic part of the molecule. There's nowhere here for water to interact. There's no charges, there's no partial charges. Water just can't get a foothold here at all. Then on the head of the molecule, the situation changes and it's hydrophilic. You have glycerol that has some polar bonds between oxygen and carbon. You also have a phosphate group which has an actual full charge on it. And then in the organic molecule, you have more polar bonds between nitrogen and carbon, and this nitrogen in choline has a charge. So, this organic molecule at the top could vary between phospholipid to phospholipid, but it's generally at least going to have polar bonds, if not an actual charge on it as well. So anytime you have a molecule that's like this, that has one part of it that is hydrophilic and then another part of it that is hydrophobic, we have a special name for that type of molecule. It's called amphipathic. So an amphipathic molecule always has one part that can dissolve in water or interact with water, and another part that cannot. It's always going to have one part that is either polar or charged and is hydrophilic, and another part that is nonpolar and hydrophobic. Anytime you have an amphipathic molecule and you put it into water, if you just wait, it's going to spontaneously form little structures. It'll either form just a little ball, or it could even form a bubble, which would be sort of like a cell membrane. The most common example of an amphipathic molecule that we're used to seeing is just soap. All soaps are going to be amphipathic. That's kind of the way that they work. They have one part of them that's able to interact with water and another part that's able to interact with lipids because it's hydrophobic. So the hydrophobic part is going to kind of pick up grease on your dishes, or whatever it is that you're cleaning, and then once you kind of rinse it off, the hydrophilic part is going to interact with the water and be washed away, and it'll end up carrying with it the grease that it grabbed with its hydrophobic part. And of course, when you are using soap in water, you get suds. So those are just, the little bubbles that are spontaneously forming anytime you add an amphipathic molecule to water. So here we have some kind of images of those little structures that are spontaneously formed in water by amphipathic molecules. The little ball is actually called a micelle if we're trying to be fancy about it. You can see the diagram of a micelle right here. So the balls here are the heads of, in this case, phospholipids, or whatever amphipathic molecule this would be, and then you have tails coming off of it. The tails are always hydrophobic and the heads are hydrophilic. So it's just going to spontaneously arrange itself so that the tails are all kind of pointing towards each other. That way they're not pointing towards water because they can't interact with water. But water loves to interact with the head part. So you're just going to kind of inevitably have the tails ending up being excluded by the water, and they end up being kind of forced together into this little ball. And once you have them in this ball, there's no water at the inside of this ball. Water is only on the outside, and it's interacting with the heads of each one of these molecules. That interaction between water and the heads is kind of stabilizing this molecule, or I should say stabilizing this structure, this micelle structure. Also the fact that you have no water on the inside and the hydrophobic tails are all able to interact with each other, that also kind of stabilizes the structure. Anytime you have an amphipathic molecule in water, it's always going to form structures in which the hydrophobic part of the molecule is kind of hidden from the water, and the hydrophilic part is actually facing the water and interacting with the water. So that's the simplest structure that you could form, the micelle. But you could also form more like a bubble, which we would call a lipid bilayer. So in the lipid bilayer, you have your lipids, your amphipathic molecules, lining up with each other to form two sheets. And the sheets are always going to be pointed in opposite directions, or kind of oriented in opposite directions. So on the first sheet, the heads are up and the tails are down. On the second sheet, the tails are up and the heads are down. That way, you have the tails pointing to each other, and these are the hydrophobic parts. So they like to be pointing to each other, because when they're all close together like this and pointing to each other, you don't have any water in the middle of this structure. And they don't like to interact with water. Water doesn't like to interact with them. So water, will prefer to be kind of excluded from this area, and here, it is. And these tails are just kind of free to interact with each other now. Water will be interacting with the heads of these amphipathic molecules on either side. So on either side of the lipid bilayer, you have this kind of sheet of heads that are hydrophilic and that water is interacting with, which is going to stabilize that structure. So the plasma membrane in cells is actually an example of a lipid bilayer. It's a phospholipid bilayer because the specific type of lipid that makes it up is the phospholipid. But you also have in eukaryotic cells a lot of other structures that have membranes around them, and we're going to talk about those in our next chapter, actually, chapter 7. We call them organelles. They're like tiny organs that do, different stuff and are just kind of wrapped up with a membrane. Those membranes are also just phospholipid bilayers. And also sometimes you'll have a really small phospholipid bilayer inside a cell, and we call that a vesicle. And cells might use that to kind of store things or transport things within the cell. You can also make them in the lab. So just to kind of go over the structure one more time, the cell membrane, also called the plasma membrane, is shown here. You can see that it's a bilayer, or what we call a lipid or phospholipid bilayer. The orange parts are the heads of these molecules, and then the yellow parts are the tails. So the tails are all facing towards each other. The heads are facing towards the water on either side of the bilayer, and they're interacting with that water. You don't have any water in the middle of the membrane where the tails are, which would be the hydrophobic part. If we zoom in to the actual molecules, we see they might look something like this. They're going to be phospholipids. You have 2 fatty acids or hydrocarbons in the molecules, or in the phospholipids, and then you also have a glycerol that both of those fatty acids are attached to. And then also attached to that glycerol, you have a phosphate group with a charge on it, and then the R here is representing the other organic molecule that would be present on the phospholipid. Maybe it's a choline, maybe it's something else. It's at least polar, but maybe it's charged as well. So this head part has a lot of charges, or has some charges, and it has a good amount of polar bonds, so it's hydrophilic and can interact with water. The tail part is hydrophobic. It has almost exclusively nonpolar bonds. It cannot interact with water. So anytime that you have a cell enclosed by a membrane, the cell is going to need to be able to transport stuff across that membrane. So of course it needs to get nutrients from the environment and bring them into the cell. It also is going to produce some waste products that it needs to transport outside of the cell. At the same time as the cell needs to transport things across the membrane, it also needs to prevent other things from moving across the membrane. So once the nutrients get into the cell needs to be able to kind of keep them there and not let them kind of leak back out through the membrane. Also just kind of molecules and structures that are in the cell, they need to stay in the cell. You can't have them just crossing the membrane and leaving. Waste products need to be transported out of the cell, but once they're out, you need to keep them out. And also if there's any toxins in the environment, you need to keep those out as well. So the cell needs to be able to kind of control what things are crossing its membrane and how they're crossing it. Every substance is going to have some kind of inherent ability to cross the membrane or an inherent disability to cross the membrane. So I guess every substance will have it'll either be kind of naturally easy for it to go across the membrane, or it'll be kind of naturally difficult for it to go across the membrane. And we describe that using this term membrane permeability. So the permeability of a membrane is referring to which substances are kind of naturally able to cross it and how quickly they can cross, or kind of how well or how easily they can cross. The permeability of any given substance is how easily it is able to cross a membrane. So you can either talk about the permeability of a substance, or you can talk about the permeability of a membrane to multiple substances. Every membrane is going to be mostly its hydrophobic interior. So as you can see in this diagram, you have, a layer of heads, of phospholipid heads on either side of the membrane, and in the middle you have your 2 layers of tails. And the tails are occupying most of the volume here. The heads are smaller and the tails are kind of long. So most of the membrane by volume is this kind of hydrophobic tail region. And that is going to be kind of controlling what is able to cross the membrane naturally. So anything that is nonpolar and small will be permeable, which means it's easily able to cross that membrane. Anything that is large or charged is impermeable, which means that it cannot cross the membrane, or maybe it can a tiny bit very slowly. And then you have kind of the stuff that's in the middle. So if you have a polar molecule, if it's large, it's going to be impermeable. If it's small, it might be like slightly permeable or moderately permeable. So it will be able to cross the membrane maybe like somewhat or a medium amount. And if you have a large molecule, usually that's going to be impermeable or non-permeable, but it might still be permeable, at least to some extent, if it's also non-polar. So this diagram is kind of showing you different examples of different substances that have different membrane permeabilities. So at the top here, our highest permeability stuff is going to be stuff like oxygen, carbon dioxide gas, and nitrogen. So these molecules are nonpolar, and they're very, very small. So nitrogen, well, molecular nitrogen and molecular oxygen, and each of those molecules is just two oxygens or two nitrogens. So of course nitrogen has the same electronegativity as nitrogen, and the same thing for oxygen. So those are going to form nonpolar covalent bonds with each other. In carbon dioxide, when you have carbon attached to an oxygen, that is a polar bond, but it's kind of mirrored by the same thing on the other side. So carbon dioxide is a straight molecule that has like an oxygen on either side, and then in the middle a carbon, and on the other side another oxygen. So it's straight, which means that when you look at the whole molecule, the charge distribution is even across it, or it's symmetrical. Because it's symmetrical, it ends up being a nonpolar molecule, even though it has polar bonds in it. And these are all very, very small molecules. So they're very easily able to cross the membrane. They can just kind of wander through this hydrophobic region because they themselves are nonpolar and hydrophobic. And they'll just kind of come out the other side. They can just diffuse right across it. In kind of the middle category, we have stuff like water and glycerol. So water is a very small molecule, but it's also very polar. So it can cross the membrane because it's so small, but it doesn't cross very quickly because it's so polar. So its arrow is going to be a little bit narrower because it's not as able to cross. And some of it is going to not cross. It's going to just kind of bounce back off. So water is only able to cross the membrane kind of like a medium amount, or it has a moderate permeability. Then you also have glycerol, which is way bigger than water is. It's still a pretty small molecule, but compared to water, it's pretty huge. And it also has several polar bonds in it. It has a bunch of nonpolar bonds, but it has several polar bonds. So it's somewhat polar, especially because those polar bonds are all like together on the same side of the molecule. So the entire molecule is polar, and it's kind of a bit bigger. So it's able to cross somewhat. It's not as polar as water, so it's able to cross somewhat, but also some of it is not able to cross. Then going further down the permeability kind of ladder, we get to glucose and sucrose, which are both monosaccharides. They are polar molecules, and they're also kind of big. They're way bigger than glycerol is. So because they're so big and kind of polar, that makes it pretty hard for them to go across. They can cross a tiny, tiny bit, but most of them just kind of bounce off the membrane and end up, on the same side that it started on. And at the very bottom, we've got our ions. Chloride, potassium, and sodium ions. So these things have full charges on them. They're very, very small, but they're very charged, and they're not able to cross the membrane really at all because of that charge. It's really hard for that charge to kind of meander across the middle region of the molecule that is all the middle region of the membrane that is all hydrophobic. So sometimes the cell needs something to cross the membrane, even though it is impermeable. And when that is the case, the membrane is going to need to use, or I should say the cell is going to need to use a special type of protein to help that non-permeable substance get across the membrane. Also for things that are kind of moderately permeable, like water, like it can cross somewhat, but sometimes the cell needs a lot of water to cross, not just a little bit. And so in that case, the cell needs to use a protein to help the water get across the membrane. And we'll talk about the types of proteins that are used in a moment. So different membranes will have different permeabilities, kind of depending on the chemical characteristics of the molecules or the lipids that make them up. So the phospholipids or the cholesterol. So the membrane is mostly composed of phospholipids, of course, and there's a bunch of different types of phospholipids. And one way that phospholipids can differ from each other is in which fatty acids or which hydrocarbons are attached to the glycerol and forming the tail. So you could have some fatty acids in the tail that are long, or some that are short, some that are saturated, some that are unsaturated, and all those characteristics are going to affect the membrane permeability. So the length of the phospholipid tails and whether or not they're saturated, or kind of how saturated they are, is going to affect the permeability of the membrane. And then also whether or not you have cholesterol, and kind of just how much cholesterol you have in the membrane is going to affect its permeability. So first, focusing on the phospholipid tails, if you have unsaturated tails or short tails, then you're going to end up having a higher membrane permeability. So that's kind of shown in this diagram here. When the tails are shorter, the membrane itself is going to be thinner. And of course, it's just easier to get across the membrane that's thinner than a membrane that's thicker. When the fatty acids on the phospholipids are unsaturated, or at least when some of them are unsaturated, that means that you have these bends in them. Right, an unsaturated fatty acid will always have at least one bend in the molecule, where if it has a double bond, it's going to have a bend. And those bends prevent the phospholipids from kind of packing very closely together. So they kind of end up a bit more spaced apart when they have unsaturated fatty acids in them. And the fact that they're more spaced apart, of course, just makes it easier to get between them. On the other hand, if you had phospholipids in your membrane that have very long tails and that are saturated, that's going to decrease the membrane permeability and make it harder for things to get across. Of course, when the tail is longer, then the whole membrane just ends up being thicker, and it's harder to cross something that's thicker than something that's thinner. Also, when all of the fatty acids in there are saturated, that means that they're all perfectly straight. And since they're perfectly straight, it's really easy to stack them together really closely. So they're going to be really tightly packed if they're all saturated. And it's just harder to get through, these tails when they're really closely packed together versus when they're more spread apart. So when you have those long tails on your phospholipids and saturated tails on the phospholipids, that makes for a less permeable membrane. It also makes for a stiffer membrane that's just kind of harder to, it's less flexible, harder to move the membrane or bend it, harder to kind of move things around in the membrane. And the same, or kind of, I guess I should say the opposite, for membranes that are made of phospholipids that have unsaturated tails or short tails. So the membrane permeability is going to go up for those membranes, and also the stiffness is going to go down. So it's going to be a more flexible membrane that's easier to move stuff around with. And then we had mentioned that cholesterol also affects the membrane permeability. So in all animal cells, you have cholesterol in their membranes. If we're talking about a plant cell or a bacterial cell, it doesn't have cholesterol in the membrane, but it has another type of steroid that's related to cholesterol in its membrane. So it'll either be cholesterol or something kind of related to cholesterol in the membrane. And the cholesterol is just kind of stuck in between the phospholipids. So you can see in this diagram here, you've got cholesterol stuck in between some of these phospholipids. It's always going to be oriented in this particular way. On each of these cholesterol molecules, this end here is actually polar. And so that end is going to be kind of anchored in with the heads, the polar heads or hydrophilic heads of the phospholipids, and then the rest of the cholesterol molecule, like the whole rest of it, is all nonpolar. So that's all going to be hanging out between the tails with the hydrophobic tails because it's also hydrophobic. So this little polar nub on the cholesterol is going to be kind of what's anchoring it in amongst the heads of the phospholipids. When you have cholesterol in the membrane, when you have kind of a lot of cholesterol in the membrane, it ends up kind of forcing the tails to be closer together. So you just have this bulky molecule in between these tails kind of filling up space, and that is kind of pushing the tails closer together. When you're pushing the tails closer together like that, you're going to decrease the permeability of the membrane and increase its stiffness. So when you have more cholesterol in the membrane gets less permeable and stiffer. And there's kind of one more factor that affects membrane permeability, which is the temperature. So temperature is just the average movement speed of molecules, or the average kinetic energy of molecules. So when temperature goes up, molecules are moving faster. When it goes down, molecules are moving more slowly. So you might figure that when temperature goes up, since molecules are moving faster, that would mean the membrane is more permeable. It's easier to get across it if everything is moving faster. And that is correct. When the temperature goes up, the membrane permeability goes up. When the temperature goes down, the membrane permeability goes down and the membrane becomes more stiff. So there's also a concept called fluidity or membrane fluidity, which refers to the ability of phospholipids to kind of shuffle around within the membrane. So phospholipids are not going to stay like put at where they're at in the membrane. They're going to kind of wander around. And that's what's shown in this diagram here. So we've got we have like one phospholipid here that's highlighted in purple, and the arrow shows it's kind of meandering around. It goes over here for a bit, moves over here, then over here, then over here. So these molecules are just going to be kind of all migrating around in the membrane and kind of jostling around in that membrane. How quickly they move is going to depend in large part on the temperature. It also depends on the chemical characteristics of the membrane, but if you have a stiffer membrane, they're going to move slower. If you have a less stiff membrane or a looser membrane, they're going to move more quickly. Whenever they're moving more quickly, we say that the membrane is more fluid. So when the temperature is going up, molecules are moving faster. That means those phospholipids are going to be migrating around the membrane more quickly. So the membrane fluidity is going to go up as the temperature goes up. Whenever you have those phospholipids wandering around kind of a lot in the membrane, if the membrane is like very fluid, then they end up being less tightly packed. There's only so tightly packed they could be if they're also moving past each other constantly. So, when you have more fluidity, you have less tight packing of the tails of the phospholipids, and that's the reason why it's easier to go across the membrane if the temperature is higher. It's just because basically the membrane is looser and more fluid and less tightly packed. When the temperature is dropping, the opposite thing happens. You're going to have those phospholipids moving around a lot less. Everything will be moving around a lot less when the temperature is low, so the membrane fluidity is going to go down, the stiffness will go up. The membrane is going to end up being packed tighter, or the phospholipids in there will be packed more tightly because they're less fluid, and that means that the permeability is going to go down. And actually, as the temperature continues to drop, once you reach the freezing point, membranes can actually freeze. And when the membrane is frozen, it's kind of impossible for things to go across it, and that's really bad for cells. They always need stuff to be able to go across the membrane. They're always going to need to get nutrients in and waste products out. So it's important for cells to have a way to stop their membranes from getting too stiff and even freezing if the temperature drops too low. And that is basically the reason why they have cholesterol in their membrane. So when we're at kind of normal temperatures, having more cholesterol makes the membrane less permeable because it forces the phospholipid tails closer together. But when you're at a low temperature, then the tails are trying to get as close together as possible and kind of pack in as tightly as possible before freezing. And the cholesterol is just kind of getting in the way. There's only so close together those tails could get if you have cholesterol, in between them, blocking them. And so the cholesterol is going to actually help maintain the fluidity and permeability of the membrane at very low temperatures. So at regular temperatures, more cholesterol means a stiffer, less permeable membrane. At low temperatures, more cholesterol means a more fluid and more permeable membrane that will actually not freeze. So that's good. So I had mentioned that if a cell has an impermeable substance that the cell needs to get across the membrane, it's going to need to use a protein to help that substance get across. So that means that we have proteins in membranes, and actually not just proteins that are designed to take substances across the membrane, but actually proteins that serve a lot of different roles. There's two basic types of proteins that you can find in cell membranes: the integral membrane proteins and the peripheral proteins. Integral membrane proteins are actually stuck in the membrane, so they're going clear from one side of the membrane to the other side. Peripheral proteins are just kind of stuck onto the side of the membrane, so they might go a little bit of the way into the membrane, but they're not going to go all the way across it. They're mostly just attached to the outside of the membrane, either like the outside side or like the inside side, but not really in the middle of it. So the reason why you can have integral membrane proteins that are actually sticking all the way through the cell membrane is because some proteins are amphipathic. So just like phospholipids are amphipathic because they have one hydrophobic part and one hydrophilic part, you can have certain proteins that have a hydrophilic part and a hydrophobic part as well. Of course, the backbone of a protein is going to be hydrophilic, because it has well, it has polar bonds in it, and when you put it into water, it actually gains charges. The carboxyl group and the amino group will pick up a charge in water. And then for your side chains, some of them are charged and some of them are polar. So between the backbone and some of the side chains, you're going to have definitely hydrophilic parts of any protein. But there's also some side chains that are non-polar. So if you have a protein that has a large amount of non-polar side chains, you can actually end up with an amphipathic protein where parts of it are hydrophilic and the parts that have all of those non-polar side chains are hydrophobic. So a protein like that will have an easy time fitting into a membrane if it's also kind of, shaped right in its tertiary structure. So we have an example of that in this diagram here, which is showing a cell membrane, and then in yellow you have a protein, an integral membrane protein that is traveling clear across the membrane from one side to the other. Actually, most of the protein is in yellow, but a few little bits of it are in green. So if we were to kind of unwind that protein and straighten it out, it might look something like this. And at either end you can see we've got these green amino acids. They're green because these are either polar or charged amino acids. And then all the yellow amino acids that you have in the middle of that protein, those are all hydrophobic amino acids or non-polar amino acids. So when you kind of roll this protein up into its final structure, which in this case is actually just shown as the secondary alpha helix structure, you can see that the green hydrophilic amino acids are located at either end, and the yellow hydrophobic amino acids are basically forming the main body of the protein. And you can arrange this protein in such a way that those side chains are just sticking out on all sides of this helix. So it would actually be the hydrophobic side chains that are kind of interacting with the hydrophobic tails of the phospholipids that are around the protein. And that's kind of anchoring it into the membrane. And then these hydrophilic amino acids on either end in green, those are able to interact with the hydrophilic heads of the phospholipids and also with any water that may be around on the outside of the membrane. So between the hydrophilic ends of this protein and the hydrophobic middle, it's able to actually stay pretty well anchored inside the membrane. Its hydrophilic parts are interacting with the hydrophilic parts of the membrane, and its hydrophobic parts are interacting with the hydrophobic parts of the membrane. For any integral membrane protein, you're going to see that same basic pattern where the parts of the protein that are actually going through the membrane are hydrophobic. They have nonpolar side chains. And then the parts of the integral membrane protein that are kind of anchored onto either side of the membrane or even maybe sticking out from the membrane a bit, those are going to be hydrophilic and have mostly charged or polar amino acids. And then finally, another name for those integral membrane proteins is just a transmembrane protein to indicate that it's going all the way through the membrane. So when we were first learning about cell membranes, we didn't really know that they had proteins going through them. And one really influential idea about the structure of membranes is the fluid mosaic model of membrane structure, which was proposed in 1972 by a couple of scientists, S. John Singer and Garth Nicholson. This model is basically stating that the membrane is a mosaic of not only phospholipids but also proteins that are stuck all the way through the membrane, not just kind of attached to either side on the surface of it. So we already kind of knew at that point that there could be proteins that are attached to either side of the membrane, but no one really knew that they could go all the way through. So this model proposed that they did. And it also proposed that, components of the membrane can kind of move around. So actually, when the model was first proposed in 1972, S. John Singer and Garth Nicholson were not saying that. They were just saying that proteins can go all the way through the membrane. But then as we studied membranes more and got more, knowledge about the membranes, we realized that actually proteins can travel around in the membrane, similar to how phospholipids can travel around in the membrane. And so the model was then updated to, state that, proteins can travel in the membrane wandering around from side to side within the membrane. So similar to how in this diagram we have phospholipids that are kind of wandering around in the membrane, any of these proteins that are embedded in the membrane, they can wander around. As the temperature increases, they're going to wander more. As the temperature decreases, they're going to wander less. And the cell has certain ways of controlling kind of how they're wandering or making sure that certain proteins can kind of stick together as they're traveling through the membrane. But proteins that are embedded in the membrane are not just, in general, they're not just fixed in place. They can actually move. Oh, right, and I guess just a little more detail on this diagram here. So it's showing, of course, the cell membrane here in kind of orange. You have your phospholipids. Then in blue, you have your proteins. These ones that are going all the way through are the integral membrane proteins. Then you have some of these that only go part of the way through. These are going to be the peripheral membrane proteins. Some of them you can see actually don't go very far into the membrane at all. And then these little chains coming off of some of these molecules in blue and in green, those are actually oligosaccharides that would be stuck onto the cell surface. So the glycoproteins are just proteins that have oligosaccharides attached to them. And some of these phospholipids that have oligosaccharides attached would be called glycolipids. And that's just used to help the cell signal to other cells which type of cell it is. So I just wanted to kind of briefly go over how it is that we figured out that there are proteins that go all the way through the membrane. And it's because it was hard to figure out because it's really hard to study the inside of a membrane. In general, if you're taking the membrane apart so that you could separate the two layers, you're going to destroy the membrane. It's not going to survive that process. So there's actually only one technique we have that can do that, and it's called freeze fracture electron microscopy. So the first thing you do in this technique is take your cell that you're trying to study and freeze it. And you have to freeze it really, really quickly so that ice crystals are not able to form, because ice crystals would also destroy the structure of the membrane. Once you've got the cell frozen, you will basically strike it to make it fracture. So you could use like a really fancy tiny little knife and a microscope to make sure you're hitting the cell so you can strike the cell. And the membrane is going to break at its weakest point, and its weakest point is always going to be just along the two layers. So in the diagram here at the top, you can see a cell that has been frozen very quickly, so no ice crystals will form, and then the researchers have struck it, and you get a little piece of the membrane fracturing off. And so you can see it's actually just the top layer of the membrane that has broken off in this point right here, which reveals the inside of the membrane. Once you've got that revealed, you can use a special type of microscopy, scanning electron microscopy or SEM, to actually take pictures of it. So scanning electron microscopy is different from regular microscopy because in regular microscopy you're using a beam of light to illuminate your specimen and magnify it. In electron microscopy you're actually using a beam of electrons. And the benefit of doing that is just that it lets you see things that are really, 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 really tiny. There's a limit to the smallest structure that you can see using a light microscope. And that limit for an electron microscope is much, much smaller. So anytime we're really trying to get pictures of structures within a cell, we're going to use an electron microscope to do it. And in the scanning electron microscope, what you're doing is just like hitting the sample with a beam of electrons, and then they bounce off, and you catch them as they bounce off, and it forms an image. So here you have an image of a fractured cell membrane that was taken using scanning electron microscopy. So you can see on this side where it's kind of more smooth, that's the actual external surface of the membrane. And the researchers had hit it right kind of at this point. So this kind of jagged line going down, that is the fracture line where this membrane fractured when the researchers hit it with a knife. And then the top layer here peeled off in this left side of the image. So this is actually revealing the internal surface of the kind of internal layer of this membrane. And you can see that it's real it's pocked. It's got pits and mounds all over it. So all those pits and mounds are actually the proteins. If you have a mound there, that's a protein that was sticking through the membrane and it's still there. If you have a pit, that is a space that has been left behind by a protein that was pulled out of the membrane because it left with the kind of the top layer of the membrane. So if you were to diagram that out, it might look something like this, where you have pits and mounds, like mounds that are left behind by proteins that are still present in this bottom half of the membrane, and then here's a pit that's left behind when another protein, another integral membrane protein, was pulled out with the top half as it came off. So this is currently the only way we have to study the internal structure of a membrane, or the inner surface of the membrane, where you just have those hydrophobic tails, pointing together, and it's basically what's the main way we first got evidence for proteins being able to travel clear through a membrane. So membrane proteins, as I mentioned, they have a lot of different functions. The primary functions are going to be signaling and membrane transport. So in signaling, that's where, cells are just trying to communicate with each other, so they're sending messages to each other. Anytime you have a cell sending a message to another cell, the cell that's going to get that message has to have a receptor for it, or it can't receive the message. So any type of message, no matter what it is, you're going to have a receptor. Usually messages are going to be chemical, so a cell would be kind of releasing a chemical, and then that chemical travels to another cell, and it's going to bind to a receptor on that cell, and at that point deliver the message. So this is how hormones work. When you release hormones in your body, like estrogen or testosterone, they're traveling in your blood to all the different cells of your body, and those cells have receptors for those hormones. If there's something wrong with the receptor so that it's not able to actually attach to the hormone, then you basically can't send the message. Also, the oligosaccharides I mentioned that are used to establish cell identity, you have to have receptors for those in order for it to actually work. So if you have a sperm cell that's approaching an egg cell and it wants to see, like, does this egg cell belong to the right species so I can fuse with it, it's going to be looking for oligosaccharides on the surface of that egg cell to indicate it's the same species as the sperm cell. And the sperm cell is going to identify those oligosaccharides by using a receptor for them. So if the egg cell has different oligosaccharides that are not, of the right species, those oligosaccharides are not going to be able to bind to the receptor that the sperm cell has, and the sperm cell will realize that, there is no compatible egg cell around for it. Those are just a couple of examples. There's actually many signaling molecules in the body, or in any organism, and they all are going to have their receptors. And usually receptors are located on the surface of the cell, so it would be a protein that's embedded in the membrane. Then for membrane transport, of course, as I had mentioned, anytime you have a substance that can't kind of cross the membrane by itself because it's impermeable, but the cell needs it to cross anyways, you need to have a protein to help that molecule cross. So basically in the rest of this presentation, or in the rest of this kind of chapter, we'll just be going into more detail with those proteins that are helping impermeable substances cross the membrane, and how it is that cells are getting things to cross their membrane. Before we get into that, I'll just kind of real briefly mention some probable characteristics that early membranes would have had. So, of course, we don't have any actual examples of the first membranes that arose in the evolution of life, so we don't really know what they were like exactly, but we've got some ideas. Probably they're not made out of phospholipids like the cell membranes that we see today. Probably they were made just out of fatty acids, so just out of these individual chains of carbon and hydrogen with a carboxyl group at the end of the chain, or else located somewhere in the chain. But the ones, the fatty acids that probably formed early membranes would have had their carboxyl group at the end of the chain, most likely. So fatty acids are amphipathic. Of course, most of them is hydrophobic, and as a whole, the molecule is hydrophobic, but there is just this tiny little patch right where you have your carboxyl group that is actually hydrophilic, because you have carbons bonded to oxygen, and an oxygen bonded to a hydrogen at that point. Of course, if you put this carboxyl into water, since it's acidic, it might lose that hydrogen, and then you would just actually have a negative charge on the oxygen. So here you have polar bonds and potentially even a charge with this carboxyl group. Which means you do have this teensy little edge of the fatty acid that is hydrophilic, where water can interact with it. So that means that it is actually possible for fatty acids to form structures like micelles or bilayers in water. When you're building a membrane out of fatty acids, it's going to be kind of maybe less stable than a membrane made out of phospholipids. A fatty acid is just smaller than a phospholipid and its structure is simpler. When you're making a membrane out of fatty acids, it's going to be like a thinner, simpler membrane kind of. And that's going to mean that it's easier for stuff to get across the membrane, especially if you have some of these unsaturated fatty acids in your membrane that are bent, and they're going to be basically, limiting how closely packed the membrane could be. So it's going to be kind of leaving little spaces in the membrane where things kind of sneak across. So if you're making a membrane out of fatty acids, basically it's going to be a more permeable membrane that allows more things to cross it. And that's going to be pretty important in the early evolution of life, especially if the RNA world hypothesis is true, because then you would not have had proteins around. You would have just had RNA and then these fatty acids maybe kind of assembling into a membrane around some ribozymes. And you're going to need a lot of things to be able to cross that membrane, but you're not going to have proteins that could help them cross if they're not permeable. So that means you need a membrane that's really permeable, that just lets a lot of things cross it. Okay, so now we'll move on to membrane transport, so how things are crossing the membrane and the different types of proteins that help them cross. So whenever you're moving a substance across the membrane, or whenever the cell is moving a substance across the membrane, there's always two basic ways to do it, either passive transport or active transport. In passive transport, the cell doesn't need to spend any energy to get the thing across, and the substance is always going to move down its concentration gradient, which means from the side where you have more of it to the side of the membrane where you have less of it. In active transport is kind of the opposite thing. The cell always needs to spend energy to do active transport, usually in the form of ATP. So active transport costs ATP, and you're always moving a substance up its concentration gradient. So you're taking it from the side of the membrane where you have less of it and moving it to the side where you have more. And then we have kind of subtypes for each of those types. So for passive transport, the subtypes of it are simple diffusion and facilitated diffusion. In simple diffusion, you have a permeable substance that is able to cross the membrane normally, and it's just going to diffuse across the membrane from the side where you have more to the side where you have less. In facilitated diffusion, you have an impermeable substance. So it is not able to cross the membrane by itself, or maybe it can cross kind of only a little bit, like water or glycerol can, but the cell needs a lot of it to cross. So this is a substance that either can't cross the membrane by itself or has some trouble doing that. And so the cell is going to have a protein that is basically going to provide a path for the substance to cross, but it still doesn't cross or cost energy. So the substance is just going to flow through this protein that's just providing a path for it from the side where you have more of the substance to the side where you have less, and it doesn't cost energy beyond, of course, building that protein path in the first place. Then you have active transport. The first type of active transport is just basically active transport. It could also be called primary active transport, but if you just hear active transport, that's usually referring to kind of the first type of active transport, where you actually have a protein that is serving as a pump, and it's going to pick up a substance from the side of the membrane where you have less of that substance, and it's going to use some ATP, and it'll drop that substance off on the side where you have more. Then you also have another type of active transport, which is called secondary active transport, and it's also called co-transport. In that type of transport, you are using ATP indirectly, and it's a little bit more complicated. So actually, we're not going to go into the details on it right now, but we'll talk about it later in the chapter. So over here, I just have diagrams summarizing those types of transport. Here you have the types of passive transport First, you have simple diffusion, where a permeable substance just diffuses across the membrane from the side where you have more of it to the side where you have less. And then over here we have facilitated diffusion. So these substances are going to be impermeable. They either can't cross the membrane or they can only cross a little bit. And then you have proteins that are providing paths for them to cross. Over here you have a protein channel. It's basically just a hollow tube that lets the substance go across, and it's just going to flow down that tube by diffusion, going from the side where there's more of it to the side where there's less. This protein works in a little bit more complicated way, but it still doesn't cost energy and is basically just providing a path for the substance to move from the side where there's more of it to the side where there's less. Then here you have active transport, kind of standard active transport or primary active transport. This is actually a protein pump. It's going to pick up substances from one side of the membrane and move them to the other. In this case, it's moving these squares. So you can see there's less squares on this side of the membrane and more on this side. So it's actually going to pick up these squares from this side of the membrane and drop them off on the other side where there's more of them already. So therefore, it's pumping them up against their concentration gradient. And it costs ATP to do that. So this pump is going to use ATP in order to move these squares across the membrane. This is actually a diagram of secondary active transport, but I'm not going to explain it right now. I'll just, when we get into the more details on secondary active transport, I'll explain it then. I just wanted to put it to kind of be complete. So passive transport is always relying on diffusion. So in order to understand passive transport better, first we need to talk about diffusion. In diffusion, you have, you're always going to have like some type of substance, and you're going to have kind of two different areas. You're going to have one area where you have a high concentration of that substance, and another area where you have a low concentration of that substance. And in diffusion, you just have spontaneous movement of that substance from the area where there's more of it to the area where there's less of it. This is always going to be taking place in solution, so the substance that we're talking about is going to be a solute. A solute is anything that is dissolved in a solvent to form a solution. The solvent is just a substance that does the dissolving. So when we're talking about organisms, the solvent is always water, right? That's just everything that's in your cells that is dissolved in water. Water is the universal solvent. It's the solvent inside your cells. So all the substances in your body, if they are something that can dissolve in water, they will dissolve in the water, and they will be solutes that can then diffuse through the water from an area where there's more of them to an area where there's less. So over here you have a basic diagram of diffusion. Here you've got just a beaker of water, and then this person has a dropper filled with food coloring, and they're just dropping it into the water. So when you first add a couple of drops of that food coloring to the water, you would see that there's just one spot in the water that's really like colored, really red maybe. So that's where all of your dye molecules are at. These dye molecules are now the solute, and the water is the solvent. If you just wait and do nothing and just let time pass, you're going to notice that the color starts spreading out. So these dye molecules are going to start moving from the area where you have a lot of them to the areas where you have a little of them, which is just the rest of the beaker. So as time passes, they're going to get more and more spread out. So they're just diffusing out through this beaker of water. If you continue letting time pass, eventually you're going to see that the whole beaker has kind of an even color. It's just the same color throughout the whole beaker. So that means that the dye molecules are actually evenly interspersed throughout the liquid. So the solute is now basically has a constant concentration throughout this liquid. There is no one part of the beaker that has more dye and another part that has less dye. It all has the same amount of dye. So when we start out here, we have a concentration gradient. We have a high concentration of dye molecules right where you put that drop, and we have a low concentration elsewhere in the beaker. The dye molecules move down their concentration gradient, which means they're moving from the place where you have a lot of them to the place where you have a little bit, which is the rest of the beaker. They continue to get more and more spread out. Eventually, when they get perfectly spread out, now there's no concentration gradient anymore. Now there's no part of this beaker that has more dye or less dye. It all has the same amount of dye. So now diffusion is going to stop. It always stops when the concentration of solute is equal kind of everywhere. So that is a spontaneous process. That's going to happen, just by itself. You don't have to put in any energy at all. You can just wait and it'll occur. You can also see this happening with tea. If you're making a glass of tea, you just put your tea bag in your water, and if you watch it, if your glass is clear, you can actually see the tea leaving the tea bag and diffusing throughout the glass of water. So you're always going to have more tea molecules right by the bag and less tea molecules in the rest of the glass. So that is a concentration gradient, and those molecules are going to move down the gradient spontaneously by diffusion until eventually, when your tea is done, there's an equal concentration of tea molecules all throughout the glass. And then I guess the final thing I'll note here is that when you get to this final state where the dye molecules are kind of equally concentrated everywhere, or your solute is equally concentrated everywhere, there is no concentration gradient anymore. It's the same concentration everywhere, and at that point we say that we're at equilibrium and no more diffusion is going to occur. So just like a solute can diffuse across a membrane, or down a concentration gradient, a solvent could also diffuse. And in biology, the solvent we always talk about is water. But we are not going to call it diffusion when water moves down its concentration gradient, because diffusion is specifically only for solutes, and water is the solvent. So we have a special name for that, and we call it osmosis. So when water is moving across a barrier, from the side where you have more water to the side where you have less water, so down its concentration gradient, that's called osmosis. But there's always going to be, like I should say, if you have more solutes in the water, you're going to have less actual water, because some of the space there is being taken up by solutes. So we don't say, when we talk about osmosis, we don't say that water moves from the area where you have more water to the area where you have less water. Instead, we say that water moves from the area where you have less solutes to the area where you have more solutes. So that's kind of illustrated in this diagram here. So you have a beaker here that's filled with water, and in the middle of the beaker you have a membrane. So that's kind of creating a barrier and dividing this beaker into two sides. The black molecules here are water, and the white molecules are sugar. So on the right side of the beaker you have nothing but water. On the left side you have a solution of water and sugar. So sugar is the solute there, and water is the solvent. This membrane is a semi-permeable membrane, which means it lets some things cross and it does not let other things cross. This particular membrane will allow water to cross. So water is permeable to this membrane. The sugar molecules are not, though. So sugar can't cross. Sugar is impermeable across this membrane. So the sugar is always going to be trapped on the left-hand side. Only the water can move. And osmosis will only occur actually if you have a situation where the water can move across a barrier, but the solute can't move. So you can actually kind of see here the difference in water concentration across the two sides. The volume of liquid on each side of the beaker is equal, but you've got a lot more black dots on the right side and a lot less on the left side. That's because some of the volume on the left side where you have the solute has been being taken up by the solute. So these sugar molecules are occupying some of the space over here, and that means there's less space left over for water molecules to occupy. So wherever you have more solutes, you're always going to have a lower concentration of water. Wherever you have less solutes, you're going to have more water. In osmosis, water is always moving from where you have more of it to where you have less of it, but that also means that water is always moving from where you have less solutes to where you have more solutes. So we always state osmosis as being the movement of water from where you have a lower concentration of solutes to where you have a higher concentration of solutes. So if you kind of just wait, you will see that water will actually end up crossing the semi-permeable membrane by osmosis, and you would get a situation maybe like what you have down here. So a lot of these water molecules have actually crossed to the left side of this beaker to kind of even out the concentration of water or the amount of water molecules on either side. So you can kind of see that like the kind of the density of water molecules is now equal on either side of the membrane after osmosis has occurred. And again, this is only going to occur if the solute molecules themselves can't move. If this membrane allowed the solute to cross, then you would just have sugar diffusing across the membrane from where you have more of it to where you have less of it until you had an equal concentration of sugar everywhere, and that would be, you know, the end of the diffusion. Since the sugar cannot cross this barrier, that means that water will move instead by osmosis. It will always move to the side where you have more solutes. So water always follows solutes. So this is going to be occurring in your cells because of course your cells are full of water, and you know, if you're a multi-celled organism that has like a body, the outside of your cells is full of water also. There's just water everywhere. So water will end up moving across the cell membrane by osmosis. So water is able to cross the cell membrane in a couple of different ways. It can cross by simple diffusion and by facilitated diffusion. It can cross only like kind of a little bit by simple diffusion. It is very small, which lets it kind of get across, but on the other hand is very polar, and that makes it hard for it to cross. So water can cross the cell membrane by simple diffusion, but it just doesn't do it very quickly. It just does it, you know, kind of slow. So you also have some water crossing the cell membrane by facilitated diffusion, in which the cell has provided a protein that's going to give the water a path to get across. And for water, the specific protein that lets water cross the membrane in facilitated diffusion is the aquaporin. So here you have a diagram kind of showing those aquaporins in blue. They're just basically providing a channel where water can cross the membrane. And water is going to be the little purple dots, so it's just going to cross by facilitated diffusion, going from the side where you have less solutes to the side where you have more solutes. So for osmosis in cells, water is always going to go to the side of the cell membrane where you have more solutes by osmosis. That ends up being really important for cells because they need to have a certain amount of water in them, but they can't have too much. So cells are going to need to control how much solutes they have inside of them compared to the amount of solutes that are outside of them. And we refer to that concept as tonicity. So tonicity is kind of how many solutes you have in a given solution compared to the concentration of solutes in a cell. If a solution is isotonic, that means that the concentration of solutes outside the cell is the same as the concentration of solutes inside the cell. In that situation, you're not going to have osmosis occur because the solute concentration is the same everywhere. In a hypertonic solution, you have more solutes in that solution outside the cell than the amount of solutes that you have inside the cell. In a hypotonic solution, it's the opposite. You have less solutes in the solution outside the cell and more solutes inside the cell. This is really important. Cells need to be in an isotonic environment. If they're in a hypertonic environment or a hypotonic environment, that means that you're going to have water either exiting or entering the cell by osmosis, and that can cause problems for the cell. If you have a hypertonic environment and there's more solutes outside the cell, since water always follows the solutes, it is going to leave the cell and go to the outside of the cell where you have the more solutes at. If you have a hypotonic solution and there's less solutes outside the cell, then water is going to be following the solutes, so it's going to be entering the cell to where you have more solutes. So here you have some kind of images of what can happen in those types of solutions, starting with a hypertonic solution where you have more solutes outside the cell. Water is always going to follow the solutes by osmosis, so it's going to leave the cells and the cells are going to shrivel up. If they get too shriveled, they're actually going to die. The cells have to have water inside of them to survive. If there's not enough water left, they will die. In the middle, you have an isotonic solution where the amount of solutes inside the cell is the same as the amount of solutes outside the cell. This is beautiful. Nothing bad will happen to the cells here. If water enters the cell, it'll just leave again. You're not going to have kind of overall movement of water into the cell or out of it by osmosis. Then finally, in a hypotonic solution, you have less solutes outside the cell and more inside the cell. So if water is following the solutes, it's going to go inside the cell, and these cells are going to swell up. If they get too big, they will actually burst, which is what's happening to this cell right here. And you know, that means that they'll die. So this concept of tonicity ends up being super important for medicine. Anytime you're giving somebody an injection, it has to be isotonic. There's like a few limited circumstances in which you would give a hypertonic or a hypotonic injection if a person has like specific medical conditions, but in general, it needs to be an isotonic solution unless they have a specific need for a different type of solution. So that's the reason why you can't just inject somebody with water. It has to be saline. Saline is basically water that has, enough salt in it to be kind of the same concentration as the inside of the cell. So saline is isotonic. If you were to inject somebody with just straight water, they're going to end up with hypotonic blood, where there's less solutes in the blood than you have inside of their red blood cells. And then their red blood cells are going to swell with water, and they can actually burst. And if that happens too much, the person can die. So that's why we care about tonicity, basically. So just to kind of review that concept again, over here we have a hypertonic solution. So if we start with a hypertonic solution, you can see there's more solute molecules outside the cell and fewer inside the cell. So if water is going to follow the solutes by osmosis, water is going to leave the cell. And that means the cell is going to end up shriveled. If it just shrivels a little bit, that's okay. If it shrivels a lot, it's going to die. Over here we have a hypotonic solution. So you can see there's fewer solutes outside the cell and more solutes inside the cell. So in this situation, water is going to enter the cell. It's always going to follow the solutes, so it's going to go to the side where there's more solutes, which is inside the cell. If you have too much water enter the cell is going to end up bursting. So if you just have a little bit of water enter the cell and it swells up a little bit, it's probably, it's okay. You can live with it anyways. But if you continue to have water enter the cell, like eventually it'll be just too much for the cell to contain and it'll burst and it'll die. In the third column, you have just an isotonic solution. So the amount of solutes outside the cell is the same as the amount of solutes inside the cell. Water is not going to be kind of moving by osmosis in any direction, and you're going to end up with no change in the size of the cell. So in an organism, organisms are always trying to maintain an isotonic environment, where there's as many solutes outside the cell as there are inside the cell. Otherwise, there's going to be problems. So finally, back to facilitated diffusion. So in that diagram we looked at for facilitated diffusion, we saw that there's two kind of types of proteins that can let substances cross. The first one is a protein channel, and the second one is a carrier protein. So first we'll talk about the protein channels. A protein channel is just an integral membrane protein that forms a hollow tube. And within that tube, you have a space for, substances to get across. So it's basically just providing a little tunnel through the membrane, and substances that can't normally cross the membrane or can only cross a little bit because they're impermeable are able to diffuse across the membrane through that little tunnel. Different protein channels for kind of different substances will have special names. So for ions, those are almost completely impermeable because they're charged. They're going to cross the membrane through ion channels. So it's a protein channel that will only allow ions to cross, and we call it an ion channel. For water, we call the channels for water aquaporins. They don't let other things cross, only water. And again, these substances are always moving by diffusion. So all the protein channel is doing is providing a space or a path for them to get across the membrane. It doesn't provide any energy or anything like that, just a way for them to go. So then the substances will always move through that protein channel by diffusion, which means they're going from the side of the membrane where you have more concentration of them to the side of the membrane where you have a lower concentration of them. Most protein channels are selective, which means they don't just allow any old thing to go across them. They only allow maybe just one thing to go across, or sometimes maybe a couple of things. So you see this for ion channels all the time. Ion channels will usually allow maybe only one ion to go across, or maybe it'll be a couple different ions. There's some protein channels that allow like cations to cross, so anything that has a positive charge can go across. But a lot of a lot of ion channels will only allow a single ion to cross. So maybe it allows potassium to cross, but sodium cannot get through. The way that the protein channel achieves that like selectivity is by its shape and by the chemistry of the amino acids that are facing into the tube. So the tube will generally be shaped so that it like kind of perfectly fits the substance that's supposed to cross it, and other things that are a different size, or I should say other things that are a bigger size, they can't get through. Also, the chemical characteristics of the amino acids that are facing into the tube will also help select for things. So if you're trying to have a tube that allows potassium to go through, but it does not allow it does not allow like chloride to go through, maybe it'll be shaped just so to fit potassium, and maybe it'll have maybe some negatively charged amino acids in there that would be attractive to the potassium since it has a positive charge. But chloride has a negative charge on it. So even if it's a similar size to potassium, it's actually going to be repelled by those negatively charged amino acids that are facing into the tube, and it's not going to be able to enter the tube. So you're usually going to have some amino acids in there that can have some type of interaction with the substance that you want to cross, and they're not going to interact with other substances. So that's kind of shown over here, where you have an aquaporin. So these are little water molecules that are traveling through the aquaporin, and right in the middle of the aquaporin you have a couple of key amino acids in here that can interact with the water just so in a way that allows that water to get across. But if you come in here and you are not a water molecule, you're not going to be able to interact with these amino acids correctly, and you're not going to be able to get across. So only water is able to pass through this aquaporin. Another thing about protein channels is that a lot of them can be gated. So some of them are just always open, but the gated ones are going to be closed at some times, and at other times they'll be open. And the cell is able to actually control when those channels are open and when they are closed. For protein channels that are gated, there's different ways to gate them. Two of the most common ways would be ligand gated, which means that a specific molecule has to bind to the channel in order for it to open. Another really common way would be voltage gated, which means that the membrane has to get to a particular voltage in order for the channel to open. Both of those types of gated channels are actually really important for the functioning of your brain and your muscles. So if you didn't have those gated channels in your brain and in your muscles, you actually wouldn't be able to like contract your muscles or have thoughts or have a functioning brain at all. In order for your neurons to signal to each other, they have to use these gated channels, especially the ones that are voltage gated. So up here you have an example of a gated channel. This is actually a voltage gated channel. So on the left side, it's in the closed position, and the membrane has a particular voltage where the outside is more positively charged and the inside is more negatively charged. So this difference in the charges across the membrane, that's the voltage of the membrane. Just like a battery has a positive side and a negative side, and the difference between the sides is the voltage of the battery, is the same for a cell membrane. So when the channel is closed, nothing can really get through it. It's this kind of gate at the bottom that's closed here. This channel is an ion channel. It's supposed to allow potassium to get through, and you do have some potassiums in here, but they can't get all the way through because the gate at the bottom is closed. If the voltage of the membrane changes because, well, maybe this is a neuron and you have some electrical signaling going on in that neuron, that can change the membrane voltage. So if it changes so that, in this case, the outside is negatively charged and the inside is positively charged, then the channel is going to open, which means that it's undergoing a conformational change. This little shift in the shape of the bottom is a conformational change, and now the bottom is opened so that stuff can get through. So you have maybe some water molecules that are trying to get through, but they can't. Only the potassiums can get through because the inside of the channel is selective and it only allows potassium to pass. The water is too big. So those gated ion channels are really important. As I mentioned, they're important in the brain and in muscle, but they're actually important, like, everywhere in the body. Um, and one example of a disease that happens because of a problem with an ion channel is cystic fibrosis. Cystic fibrosis is genetic, which means you have a particular mutation that gives you the disease. The symptoms of the disease are basically that your body is producing mucus that is way too thick. Not just mucus, but also other secretions that your body makes, they're way too thick. So this is going to give you different problems all across your body, depending on, depending on where it is. But one of the main problems that you see is that the mucus that's lining the respiratory tract is too thick, and it ends up building up and blocking the respiratory tract, and that makes it really hard to breathe. So normally you're going to have mucus coating like your airways, so that when you breathe in some particles, those particles get stuck in the mucus and they don't go kind of further down into your lungs. And then these cells that are lining your airways have these little whips on them, basically called cilia, and they're constantly beating, and they move the mucus out of your airway up, and then you swallow it. But if you have cystic fibrosis, the mucus in your airways is way too thick, and the little whips, actually, they can't move it. So it gets stuck there, or they just kind of move it really slow. So it's going to get stuck, and it's going to keep building up, and that ends up actually blocking your airways. And then, like these little particles that you're breathing in that get stuck in the mucus, some of them are actually bacteria, and now instead of ending up getting swallowed and going to die inside your stomach, which is very acidic, now those bacteria are just kind of chilling out in the mucus in your airways, and they can actually establish infections there. So the actual mutation that causes cystic fibrosis has been identified, and it's a mutation in this gene that's called the CFTR gene. The CFTR gene has the instructions to build the CFTR protein, which is an integral membrane protein, and it's actually an ion channel for chloride. So, the way that it functions is it allows chloride ions to cross the membrane. So normally, wherever in your body you're kind of producing mucus, the cells that are lining those surfaces that the mucus goes out onto, those cells are going to have the CFTR protein in them. So in the membranes, you're going to have that chloride channel, and it allows chloride to leave the cells and go out onto the surface. And on that surface, that's the same surface where you're producing mucus. So maybe this is your airways, and so you're producing mucus that lines your airways, and also those cells that line your airways have this CFTR chloride channel, and they're allowing chloride to leave the cell and go out onto the surface of your airways. That means you're going to end up with a high concentration of chloride ions on the surface of those airways. And then that high concentration of chloride ions, it actually ends up being like a high solute concentration, and water is going to be drawn out of these cells that line the airways by osmosis and into the mucus. So that makes you have really thin mucus, or at least reasonably thin mucus, mucus that is mobile and good. So if you don't have that working properly, then you're going to have some problems. So in cystic fibrosis, you have a mutation to that CFTR gene, which means that the CFTR protein that you make is broken. So normally that is like a gated channel that's able to open sometimes and you'll close it other times. But when you have cystic fibrosis, the version of that CFTR chloride channel that you have isn't able to open at all. So that means that you cannot have any chloride ions like leaving those cells lining your airways and getting onto the surface of the airways. That means you're not going to have a high concentration of solutes on the surface of the airways. That means that water is not going to be drawn out of those cells that line your airways by osmosis. That means water is not going to be entering your mucus and thinning out your mucus, and you end up with, real thick mucus. Yeah, so basically just an illustration of why those ion channels matter. So now we'll talk about the other type of protein that can be involved in facilitated diffusion, which is the carrier protein. So that's the one shown right here in the diagram. It is not just as simple as like a hollow tube or a tunnel that allows substances to cross. It's a little bit more complicated, but it still allows things to cross the membrane by diffusion. impermeable substances. So basically, it's an integral membrane protein, and what it's going to do is like be open on kind of one side of the membrane, and it'll have the substance kind of come into it and grab onto it, like bind it, and then it'll undergo a conformational change so that it actually opens to the other side of the membrane, and then it drops that substance off. And this does not require ATP. So here, this carrier protein has been kind of caught in the act of dropping its substance off. So you can see it's kind of open to the inside side of the membrane. It would have actually started out open the other way, so the little triangle would have been pointing the other way, and one of these little balls can get into it and bind in the little pocket for it. Once that little ball binds in that pocket, is going to trigger a conformational change, so just a slight change in the shape of this protein, and then that little ball will get dropped off. After the conformational change, the carrier protein is going to be opened the way it's shown in this diagram, so it's open to the inside of the membrane, and then the ball can get dropped off. So this conformational change is going to be triggered by the binding of the substance to the carrier protein. It does not require ATP. So that means that this is still a type of diffusion, or a type of facilitated diffusion, even though it's a little more complicated than just a tunnel that a substance travels through. Just like with the channel protein, with a carrier protein, it actually doesn't matter like which way the substance travels, or I guess I should say the protein is able to allow substances to travel in either direction. So it's not going to only allow things to go from the outside of the membrane to the inside. It'll go either way. Which way the substance actually ends up traveling is going to depend entirely on its concentration gradient. So if this substance has a high concentration outside the cell and a low concentration inside the cell, a carrier protein or a channel protein would always carry that protein from the outside to the inside. If the concentration gradient pointed the other direction, though, if you had a lot of that substance on the inside of the cell and only a little bit on the outside, then it's going to go the other way. Then the carrier protein or the channel protein are always going to take the substance from the inside of the cell and put it on the outside. So because these proteins don't take energy to function, and because they are just always carrying the substance in whichever direction is down the concentration gradient, that means it counts as diffusion. In general, if you have a small molecule that needs to get across the membrane but is impermeable, if you're going to do that by facilitated diffusion, you would use a channel protein. But if you have kind of like a big molecule, like maybe glucose is kind of too big to fit through a little channel. So in that case, that's when you would use the carrier protein. So carrier proteins are always going to be for kind of larger molecules that are impermeable that need to cross the membrane by diffusion. And an example of a carrier protein is GLUT1, which stands for glucose transporter 1. That is a carrier protein that actually is for glucose. And so it will always carry glucose from whichever side of the cell membrane has more to whichever side has less. So here you have that GLUT1 or glucose transporter 1 protein kind of shown in action. So it starts off sort of looking like this, where it's open to the outside of the membrane. You have a lot of glucose outside the cell, you have less glucose inside the cell. So this GLUT1 protein is going to pick up some glucose from outside the cell. There's a little pocket in here where the glucose can bind. Once the glucose binds, that triggers a conformational change where the protein is going to kind of close up on the outside and then it'll reopen on the inside. So it's going to basically switch the direction that it's open onto. Now it's open onto the inside of the cell. And now the glucose is going to be let go and it'll just float off into the inside of the cell. Once the protein lets go, then once the glucose lets go, then this carrier protein is going to end up resetting. It'll undergo another conformational change and go back to its original position so it can pick up a new glucose. So those are our types of passive transport. Now we can get into active transport in more detail. And again, active transport is always transport in which you are moving something from the side of the cell or the side of the membrane where there's less of it to the side where there's more. which means you're moving it up its concentration gradient, and we also call that moving it against its concentration gradient because it's not kind of the natural thing that happens. Anytime you're doing that, it always costs ATP. So you could kind of imagine why it's going to cost ATP to move something up its concentration gradient by thinking about the glass of tea again, right? When you're making your glass of tea, you just put your tea bag in the water and you just wait, and it'll diffuse. The tea will diffuse out of that tea bag and into your into your glass. You don't have to do anything extra. It'll just happen by itself. Technically you do heat the water up to make it happen faster, but you don't, like, if you had all the time in the world, like, it would just happen, you don't have to heat it for it to happen. Heating it just makes it go faster. So, that's diffusion. That's tea molecules moving down their concentration gradient from the area right by the tea bag where you have more of them to the area in the other part of the glass where you have less tea molecules. Once your tea is actually made, then you, if you wanted to kind of like put the tea back in the bag, that's not going to happen by itself, right? So if you just have a tea bag in a glass of tea, those tea molecules are never going to move back into that tea bag. It's not going to happen. If you were to try to make that happen, I don't know how you would be able to do that, but it's going to cost energy. That's all I know. For sure, it's going to cost some energy. So that would be more like active transport. Moving something backwards up against its concentration gradient is always going to cost energy. And it's also always going to require a protein. And the type of protein that we use for active transport is always a pump protein of some type. A pump protein is an integral membrane protein that uses ATP in order to move a substance from the side of the membrane where you have less of it to the side where you have more. So here you have an example of active transport and a pump protein. Again, moving these squares from the side of the membrane where you have less of them to the side where you have more, and using ATP to do that. Whenever this is happening and you're using ATP to kind of like activate this pump protein, that means that you're going to have this free phosphate from the ATP actually end up transferred to the pump protein. And also the energy that's associated with breaking that bond between the phosphate and the rest of the ATP molecule, that's actually also going to be transferred to the pump protein, and it will be used to power the conformational change that the pump protein needs to undergo. There's this one active transport pump that's like way more important than any other one, like by a lot, and it's called the sodium-potassium pump. It's also called the sodium-potassium ATPase. We can call it an ATPase because it breaks down ATP. So the ATPase, like the ACE part of that name, lets you know it's an enzyme. So there's part of this pump that functions as an enzyme that's actually able to strip the last phosphate group off of ATP to get energy. So technically, any protein that's able to do that, that's able to break the last phosphate group off of ATP, it would be an ATPase. The sodium-potassium ATPase uses the energy that it gets from ATP to pump sodium and potassium across the cell membrane against their concentration gradients. Every single cell in your body has the sodium-potassium pump. So when I say on there that it's found in all cells, I don't mean literally every cell on the planet that exists, not necessarily in all bacterial cells will you find this pump, but in every mammalian cell you will find this pump. Actually, in every animal cell, you will find this pump. In your body, about 30% of all the ATP that is used is just going to power this pump. So 30% of all, like, the energy that you're producing is for this pump. This is by far and away the single most expensive protein that you have. And as you might imagine, it's real important. If it was not real important, you would not be spending 30% of all your energy on it. But its job seems really simple. All it does is take three sodium ions and put them out of the cell, and then take 2 potassium ions and put them into the cell. So in your cells, you're always going to have a high concentration of sodium outside the cell and a high concentration of potassium inside the cell. So when you're taking sodium ions from the inside of the cell to the outside of the cell, that means you're pumping them up against their concentration gradient because you have more sodium on the outside. For potassium, it's the other way around. you have more potassium on the inside. So, when you're taking potassium from the outside and putting it inside, then you're pumping it up its concentration gradient. And here you have basically a drawing of the sodium-potassium pump. So this is kind of a summary of the operation of the sodium-potassium pump. So you're going to start out with this pump kind of open towards the inside of the cell. You have a lot of sodium outside the cell, but not a lot inside. So on the inside of the cell, you have the pump open like this, and it's got these 3 little pockets that are designed to perfectly fit sodium ions. So you're going to have 3 sodium ions come and grab these little pockets. Then you're going to have the ATPase function of this pump come into play. ATP is going to bind to the pump. The pump is going to catalyze a reaction that breaks off this last phosphate group from the ATP. And when it does that, it's going to use the energy of that broken bond to undergo a conformational change. So over here, you have that conformational change occurring. This is the phosphate group that was broken off of the ATP. What you have left is a DP, 2 phosphates on it. The pump is changing its shape. It's closing up on the side that's inside the cell. On the side that's outside the cell, it's opening up, and it's going to release its sodium ions. So now these sodium ions are going to just float out to the outside of the cell. And the outside of the cell is where you have a higher concentration of sodium ions. So these have now been pumped up against their concentration gradient. Now the pump is open to the outside of the cell. And potassium, a couple of potassium ions are going to come in, and there's a couple little pockets in here that are specially shaped for potassium ions to bind. So these potassium ions are going to come in and bind that little pocket. When they bind the pocket, that's going to trigger a conformational change, and that's going to kind of reset the shape of this pump. So now it's going to close on the outside and reopen on the inside. So now it's open on the inside, and these potassium ions can float out into the inside. And the inside is always where you have more potassium, so that means you've just pumped 2 potassiums up against their concentration gradient. And at the same time as that conformational change is happening, that phosphate group that was attached to our sodium-potassium pump is going to leave. So at this point, once the potassium ions leave as well, now you've kind of reset the pump and you're ready for a new cycle to begin. So the basic point of all this is to make sure that you have those concentration gradients for sodium and potassium. Right? So you always should have a lot of sodium outside the cell and a lot of potassium inside the cell. You have that because of the operation of this pump. Anytime sodium ends up inside the cell, the sodium-potassium pump will put it outside. Anytime potassium ends up outside the cell, the sodium-potassium pump will put it inside. That makes sure you always have those concentration gradients. Those concentration gradients are really important for a lot of different processes in the cell. And also, you end up putting like more ions out than what you take in. It's 3 sodium ions that will leave the cell every time, and only 2 potassium ions that enter, which means that overall the amount of solutes that you're putting out is bigger than the amount of solutes that you're taking in, and that's also important. And those are ions that have a charge. So you're taking positively charged sodiums and putting them out, you're taking positively charged potassiums and putting them in, but you're only putting 2 potassiums in for every 3 sodiums you put out. So you're ending up putting more positive charges outside the cell and less positive charges inside the cell. The fact that you are ending up with more positive charges outside and less positive charges inside gives you a voltage. That means that the amount of positive charges is not the same on both sides of the cell membrane. Anytime you have a difference in the amount of charge on 2 sides of something, that causes a voltage, just like the voltage in your battery. On one side of the battery you have positive charges, on the other side of the battery you have negative charges, so that's a big difference in charge that causes a voltage. Of course, in your cell membranes, the amount of voltage is like real little. Not nearly as big as in a battery, even if it's a small battery, but there is still a voltage across the cell membrane, and that ends up being important. It's super duper important for your neurons and for your muscle cells. They are completely unable to function without that voltage. They have to have it. your neurons have to have that voltage across the membrane in order to send messages to each other so you can think, and your muscles have to have that voltage across the cell membrane so they can contract. So that's one thing that matters about the sodium potassium pump, that it establishes that voltage across the cell membrane. And kind of between that voltage and the ion gradients, the concentration gradients that you're establishing, you're actually basically treating the cell like it's a battery. So the voltage across the cell membrane has energy stored in it. The concentration gradients for sodium and potassium have energy stored in them. So it's almost like you're taking energy that was stored in ATP and using it with the sodium potassium pump to convert that energy into a voltage across the cell membrane and into concentration gradients for sodium and potassium that also store energy. The energy that's stored in the concentration gradients for sodium and potassium is going to be used in secondary active transport. The energy that's stored in the voltage across the membrane is going to be used to contract muscle cells and to send messages in neurons. Other cells will also use it for other things, but the most important usages would be for the muscles and the neurons. This diagram here is just kind of showing how the pump operates and its effects on ion concentrations and membrane voltage. So in the middle here, this is your sodium potassium pump. It's using ATP, it's taking 2 potassiums and pumping them into the cell, it's taking 3 sodiums and pumping them out of the cell. So you also have some other channels, some other ion channels in the cell membrane that allow sodium or potassium to pass. So you end up having, you have a lot of potassium inside the cell, but it's also going to be kind of leaving the cell sometimes through these other ion channels for it. Same thing for sodium. You have a lot of that outside of the cell. It's these kind of bluish, purplish little balls. But you also have some ion channels that will allow sodium across the membrane. So some sodium will be entering the cell. So you have to have the sodium potassium pump to make sure that those concentration gradients stay intact. If you always have your potassium just kind of leaving the cell and nothing to put it back, you're going to end up draining away your concentration gradient for potassium until the amount of potassium on the inside is the same as on the outside. The sodium potassium pump is making sure that you're always putting potassium back inside the cell. Every time it leaves the cell, you put it back. And the same for sodium. Every time it enters a cell, you put it back outside. You also end up having like only 2 positive charges enter for every 3 positive charges that leave. That helps you establish the membrane potential, where the outside of the membrane is a little bit more positively charged compared to the inside of the membrane, which is a little bit more negatively charged. It's not actually like negatively charged, but it has less positive charges than the outside, so the outside is more positive. So I kind of mentioned that the sodium potassium pump establishes concentration gradients for sodium and potassium and establishes the voltage across the cell membrane. There's also one more thing it does that's super duper important. And it's all related to osmosis. So, basically cells have a lot of solutes in them. They've got a lot of stuff inside of there. Not only did they have the macromolecules that they have, of course, but they also have a lot of tiny molecules like ATP or glucose, electron carriers, cofactors, enzymes just floating around. There's a bunch of little stuff, ions of course, a bunch of little stuff inside your cells. They are full of solutes. In general, if you have a cell kind of floating around, it's probably in a hypotonic solution. Usually, solutions don't have nearly as many solutes in them as what a cell has. And so that's a problem for cells because that means that they might end up swelling up and popping. Most forms of life have cell walls to solve that problem. So bacteria, archaea, fungi, and plants all have cell walls around their cells. If they are in a hypotonic environment, which they usually are, water is going to enter the cell by osmosis and the cell is going to swell up, but it can kind of only get as big as its cell wall, can't get bigger than the cell wall because the cell wall is really strong and it's not going to expand. So they're kind of prevented from popping. If you're an animal cell though, you don't have a cell wall. So if you start swelling up with water, you're going to swell and swell and swell until you do pop. And the way that animal cells prevent that from happening to themselves is by using the sodium potassium pump. which we're going to go into that on the next slide. But first, just this diagram here is just kind of showing different cells in, different environments. So up at the top, you have an isotonic environment and a plant cell and an animal cell. Everything is groovy. Same solutes inside the cell as outside the cell. No problems at all. Down here, you have a hypotonic environment. Less solutes outside the cell, more solutes inside the cell. The animal cell is going to swell and swell and pop. The plant cell is going to start to swell, but it can't swell very much because of this really thick green cell wall that you have around it. So, it starts off with a slightly more angular shape to its cell wall. The cell wall is going to kind of bloat a tiny bit, but it's really strong. It's not going to change its shape very much. There's no way the cell is going to swell up big enough to pop. It just can't do it. The animal cell, though, not so lucky. But both types of cells, I should mention, are still vulnerable to a hypertonic solution where there's more solutes outside, and then the animal cell will shrivel up and die, and the plant cell, same thing, going to shrivel up and die. But hypertonic environments are not usually very common. They're around, but just kind of limited. Most environments are hypotonic. So back to our sodium-potassium pump and how it's helping us with our osmosis problem. The sodium-potassium pump is always putting 3 sodiums out and taking 2 potassiums in. That means that you're always putting 3 things out and 2 things in. 3 solutes go out, only 2 solutes come back in. So every time that the sodium-potassium pump operates, it's going to actually drain the solutes from inside the cell and put them outside the cell. You're going to end up having more and more solutes accumulating outside the cell, while the number of solutes inside the cell is kind of going down. It's actually sodium specifically that's leaving the cell and, accumulating outside the cell. So if you kind of start off with an animal cell that is in a hypotonic environment with less solutes outside the cell, and then you start running the sodium-potassium pump, you're going to end up putting solutes outside the cell, increasing the amount of solutes outside the cell. Actually, you're going to do that until the amount of solutes outside the cell equals the amount of solutes inside the cell. So the sodium-potassium pump is basically taking hypotonic solutions that our cells find themselves in and converting them into isotonic solutions that are non-toxic to us. We are really relying on the sodium-potassium pump to prevent our cells from, ending up in a hypotonic environment, swelling up and popping. Finally, we'll talk about secondary active transport, or also called co-transport. This is also highly related to the sodium-potassium pump. So I had said that in secondary active transport, you use ATP indirectly to pump something up against its concentration gradient. The way that you're using ATP indirectly is basically that the sodium-potassium pump is actually using the ATP, it's converting that energy in the ATP into concentration gradients for sodium and potassium, and then other proteins can use those concentration gradients for sodium and potassium and use the energy that's stored in those gradients to move something else against its concentration gradient. So in secondary active transport, you have one protein that's a pump protein. It's probably the sodium-potassium pump. It is using ATP to pump something up against its concentration gradient and establish a strong concentration gradient for that thing. Then you have a second protein called a co-transporter that is going to exploit that concentration gradient and use it to pump something else. So that's shown in this diagram here. On the left-hand side, we have our sodium-potassium pump. It's moving two potassiums into the cell at the same time as moving 3 sodiums out of the cell, and it's using ATP. So it's establishing concentration gradients for sodium and potassium, where you have a lot of sodium outside the cell and a lot of potassium inside the cell. In this case, we're going to be using this concentration gradient for sodium. So over here on the right, you have a co-transporter. This co-transporter is going to allow sodium ions to flow down their concentration gradient into the cell. It's going to use the energy of their movement to pump something else up against its concentration gradient, in this case a glucose molecule. So this glucose is being pumped from the area where you have less glucose outside the cell to the area where you have more glucose inside the cell. If you didn't have this concentration gradient for sodium where there's a lot outside and only a little bit inside, you would not be able to run this cotransporter. The cotransporter is relying on the energy of sodium diffusing down its concentration gradient in order to pump glucose. And there is that concentration gradient for sodium that allows it to diffuse down into the cell only because of the sodium-potassium pump that used ATP to put the sodium outside of the cell and establish that concentration gradient in the first place. So this particular cotransporter is actually the sodium glucose cotransporter, also called the SGLT protein. It's a cotransporter transporting sodium and glucose, allowing sodium to flow into the cell down its concentration gradient by diffusion, harnessing the energy to pump glucose also into the cell, but up its concentration gradient. And again, the sodium-potassium pump is the one actually spending the energy. It's the one actually using the ATP to establish those concentration gradients. So finally, I'll conclude by just kind of reviewing the importance, the very, very great importance of the sodium-potassium pump. Its basic job is just to take 3 sodiums and put them out of the cell, take 2 potassiums and put them in. So it's basically just establishing concentration gradients for sodium and potassium where you have a lot of sodium outside the cell, only a little bit inside the cell, and you have a lot of potassium inside the cell and only a little bit outside of the cell. In that process, you also end up establishing a voltage across the cell membrane because you're putting 3 positive charges out for every 2 that you're putting in, and you're also establishing an isotonic environment for the cell to live in because you're putting 3 solutes out and only taking 2 in. So you're putting more solutes out than what you take in. That ends up being so important. Those ion concentration gradients, you have to have them for secondary active transport. You have to have them for neuronal signaling in your brain. You have to have them for muscle contraction. Also, the voltage that's established by the sodium-potassium pump, you have to have it for neuronal signaling and for muscle contraction. And of course, one of the muscles in your body that you have is your heart, which you need. So if you don't have the sodium-potassium pump, or if it stops working for some reason, your neurons are not going to work. They're not going to send messages to each other anymore. You're going to be abruptly brain dead. Your heart is not going to continue beating. Without that voltage across the cell membrane and without the ion concentration gradients, it will not be able to beat. Your other muscles will not be able to contract as well. And that includes the muscles that allow you to breathe. So you're going to be completely paralyzed, not breathing, no beating heart. Oh yeah, and your cells are all going to pop because they're not going to be in an isotonic environment anymore. It'll go back to a hypotonic environment and they will all pop. So, the only question is which one of these things is going to kill you first if you lose that sodium-potassium pump. It's real big important.